Hello, today we're going to be talking about action potential. Action potential is pretty much how a nerve impulse travels along the axon. So it's a pretty simple process that we're going to get into today. There's a lot of different kind of background and vocabulary that we have to get through first. So we're going to start off by talking about the cell membrane. Now, the cell membrane, if you remember from bio, is selectively permeable. So things move in and out of the cell. It's composed of a lipid bilayer, and there are some channels and proteins within that cell membrane. A cell membrane is usually electrically charged or polarized. And this pretty much means inside the membrane is negatively charged. So the concentration of potassium ions is approximately 30 times higher inside the cells. Outside of the membrane is positively charged. And that's because the concentration of sodium and chloride ions are approximately 10 to 15 times higher outside the cell than inside. So potassium is K. So a potassium ion is K plus. That is the major intracellular positive ion or cation. They can diffuse out of the cell more rapidly than sodium can diffuse into the cell. The sodium is Na, so a sodium ion is the Na plus. And these are the major extracellular positive cations. So this unequal distribution of the charges of ions on the inside and the outside of the membrane is what's going to help contribute to the action potential. It's very important in the conduction of muscle and nerve impulses. If you have any questions on the biology aspect of a cell membrane or a plasma membrane, just let me know and I could review that for you. Now, ions can diffuse between two different channels. There's two types. There's a leak channel, which is always open, so things can readily go through it without having a problem. A gated channel is either going to be opened or closed, and it is going to change depending on the needs of the cell. So in the resting state, potassium ions diffuse out of the cell through the potassium leak channels. Movement of those positively charged potassium ions out of the cell leaves the inside of the membrane more negative than the outside. Now, the plasma membrane is very impermeable to sodium ions. The nerve cells will lack that sodium leak channels, and that's why there's going to be a gated channel. So what we're trying to have you understand is action potential. This is a characteristic change in a neuron membrane polarization and return to the resting state. If you look at the diagram, the resting state is measured in negative 70 millivolts. Okay. Now, the action potential is what's going to form a nerve impulse that gets propagated along an axon. Now, it's a rapid change in polarity across the axon as the nerve impulse occurs. It's something that has to cross a threshold. So if you look at the graph again, threshold is at negative 55 millivolts. All right. Now, with that, once the stimulus gets reached and we start changing the voltage, once it hits negative 55, it continues on with the action potential. It is an all or nothing effect. Now, this action potential is necessary, again, for the nerve impulses and the muscle impulses to travel. And it does need two gated ion channels, sodium and potassium. They are going to open and close in response to a stimulus, which is the signal from another neuron. And if you could see, there's the failed initiations, they never reached the threshold. And the terms depolarization and polarization we're going to define in a minute, but if a memory becomes less negative, so it goes more to the positive, we would call that depolarized or depolarization. And you can see it's from negative 70 to about plus 40. And that is going to happen because of the sudden entrance of sodium ions coming in. The reduction in that membrane voltage causes the decrease in polarity between the two sides of the membrane. Okay. Now the repolarization is when it goes now towards the negative end. Hyperpolarized means that it goes more negative. So from the resting state, it's going to travel to be more negative than the negative 70. Now, the sodium potassium pump is very important. It's an active transport system in the plasma membrane that pumps 3 sodium ions out of the axon and 2 potassium ions into the axon. Now, it's always working, but it does need energy. So if you look at the diagram, it's going to show you that the information that you're getting here, the sodium potassium pump is going to take 3 sodium ions in, and it's going to have a change in the shape, and it does need the ATP. Once that happens, they are going to be released out of the cell, and that's going to cause the potassium ions to flow in. Now, there's 3 sodium ions that are leaving and 2 potassium ions that are entering into the cell. So it's a very big difference of 3 sodium to 2 potassium. Now, there's the term potential difference. A potential difference is the difference in electrical charge between 2 regions. The resting potential is the potential difference between the region inside the membrane and the region outside the membrane when the cell is at rest. So the resting potential is when the axon is not conducting an impulse. The resting membrane potential is -7 millivolts. This is a 70 millivolt difference between the inside to the outside of the cell. Now, the negative value indicates the negativity of the inside of the cell relative to the outside. And again, this is due to the outward diffusion of potassium ions. So again, it's going to be negative inside the cell to outside. And again, the sodium potassium pump is going to help restore this. There are different terms that was shown on the graph. So I want to kind of go through that just so you can write that down. Hyperpolarized means if the membrane potential becomes more negative. And again, that was that big dip in the graph. Depolarized means that the membrane potential becomes less negative. So the inside of the neuron becomes positive because of the sudden entrance of sodium ions. The reduction in the membrane voltage causes a decrease in the polarity between the two sides of the membrane. And again, we are going to have the 3 sodium. Now, threshold potential is a certain point that the membrane will get depolarized. And once it happens, the action potential has to go on. So threshold potential is -55, and that is the graded or proportional to the intensity of the stimulation. Okay. So just showing you a little bit about how this is working. Okay. So the sodium ions 3 are going to go in. That's called depolarization. When the 2 potassium leave, that's called the repolarization. Hyperpolarization is when it goes further into the past the resting states. You can see here that this is occurring in an axon. Just to kind of remind you, the cell body has the nucleus and pretty much all the parts of the cell. The dendrites are the things that are going to be receiving the stimuli, and the axons are going to be where the impulse is going to travel along. Okay, so as far as this goes, we have a term called saltatory contraction. This means Latin to leap. The action potential is going to jump from node to node. So impulses are able to travel along a myelinated axon at speeds of up to 120 meters per second. This is 20 times faster than the speed impulses of an unmyelinated neuron at that same diameter. So you can see how that nerve impulse has to jump. Okay, and it is going to be passing over the Schwann cells and the nodes of Ronvar, the little gaps. You can just see there how the nerve impulses will be traveling to allow that action potential to take place. We say all or none response. That means if it's going to respond, it's going to respond completely. So once it hits that threshold of the negative 55, it is going to continue its action potential. So a nerve impulse is conducted whenever a stimulus or the threshold intensity or above is applied to an axon. All impulses carried on the axon are the same strength. Now, a refractory period is a brief period that occurs following a nerve impulse when a threshold stimulus will not trigger another response. It limits the frequency of impulses of a neuron. The absolute refractory period is the time when the threshold stimulus does not start another action potential. And a relative refractory period is a time when the stronger threshold stimulus can start another action potential. Okay, and this is the sodium gates are closed and the potassium are open, and it's pretty much when the repolarization is occurring. So here's just showing you that difference as far as the different refractory periods. So hyperpolarization was the same part on the graph as the refractory period. And it does show you the absolute versus the relative. And again, the absolute is a time when the threshold stimulus does not start another action potential, because that's when the nerve impulse is traveling. The action potential is started. And you can see it's there within that 2 millisecond range, excuse me. Now, the relative refractory is a time when a stronger threshold stimulus can start because we are going into that resting state. But the whole thing about a refractory period is it does give it a brief period of pause before the triggering happens. And this is just kind of showing you, again, the sodium potassium pump. I think that's a pretty important concept for you to understand. We will be doing different Pogels on this. But again, the sodium potassium pump is when 3 sodium ions are going to go in, and then the 2 potassium ions are expelled. I'll just give you a second to kind of look through that. And again, this is just showing you what's happening along the axon. It's showing you how you're at the bottom part of the graph is showing you the millivolts graph, and then the middle part is showing you this potassium and sodium, so you can see what's happening, and you can see on top that it is occurring in an axon. So just to review, an action potential is a very rapid sequence of depolarization and repolarization. It takes under a second, 1/1000th of a second. At rest, the membrane is polarized at a negative 70. Remember, the threshold was negative 55. The sodium channels open and the membrane depolarized when it goes to the more positive end toward the 0. The potassium leaves the cytoplasm and membrane repolarizes at about plus 30 millivolts. And then that brief period of hyperpolarization is when it gets past the negative 70, which would be about negative 90. And again, the threshold is reached, the sodium channels open, and the action potential goes on. As the sodium moves in, the action potential goes from negative 70 to positive 40 millivolts. The sodium gate will then close, potassium gate opens, As the potassium leaves the axon, the action potential goes from plus 40 millivolts to negative 70 millivolts. And that is pretty much how it's going to happen. Remember, the saltatory conduction is when the nerve impulse jumps over the different Schwann cells because it is myelinated. So the nodes of Ranvier is where the nerve impulse travels, jumping from node to node. Speed of this impulse is based on the diameter of the axon. The greater the diameter, the faster. And just one little fun fact, when you go to the dentist, you get Novocaine. So local anesthetics like xylocaine and Novocaine, they act by closing the gates of the ion channels in the membrane of the nerve cells. So that stops that pain to be triggered. All right. So as long as these these channels are closed, the affected neurons are unable to generate that action potential and it can't inform the brain of the pain, which is very important, at least for me when I go to the dentist. I don't know, I have very sensitive teeth, so he always has to give me 2 shots of Novocaine. And I come out of there with my face dying of numbness. So that is the action potential. If you have any questions, let me know. Just real briefly, we're going to chat about neurotransmitters because they're very important in that release of the nerve impulses. So a neurotransmitter is a biochemical that is released by synaptic vessels of an axon. There's about 50 different types that have been identified and about 100 known neurotransmitters. They may open ion channels and others may close ion channels. So we have some examples of that. We have the adrenaline, the noradrenaline, the dopamine, the serotonin, the GABA, which is gamma aminobutyric acid, acetylcholine, glutamate, endorphins. So just if you look at them, the acetylcholine excites the skeletal muscles, but inhibits the cardiomuscle gland. So it's going to decrease membrane potential. So when running, your muscles secrete this, making it easier to excite their cells. Right? We have endorphins. These bind to natural opioid receptors in the brain. It's like your runner's high. And this is going to produce emotional and also physical stress. Dopamine is involved with emotions, learning, and intention. And serotonin is involved in sleeping in particular, and also emotions and perceptions. We have norepinephrine, that is a dreaming kind of walking neurotransmitter, which is released by nerves that stimulate heart contractions. So this kind of gives you a little bit more on what they do and the different functions and what can happen if they malfunction. You can see that acetylcholine is affected with Alzheimer's disease. Your acetylcholine producing neurons deteriorate. Let's say serotonin, depression, okay? Same with norepinephrine, that happens. Dopamine is linked to schizophrenia, Parkinson's, okay? Glutamate is why you are pretty much allergic to the MSG, the monosodium glutamate. And the gamma is actually gamma aminobutyric acid. This can be linked to seizures, tremors, and insomnia. Now, these neurotransmitters can be excitatory or they can be inhibitory. The excitatory means that they increase the postsynaptic membrane permeable to the sodium ions and trigger nerve impulses. The inhibitory neurotransmitters make it less likely that a threshold will be released and lessens the chance that a nerve impulse will occur. So when an impulse reaches that end of the presynaptic neuron, it triggers an opening of the calcium ion channels in which part of the membrane. Calcium ions will flow into the synaptic knobs of the cell, which induces the exocytosis. As calcium ions flow into the cell, the membrane of the number of synaptic vesicles fuse with the plasma membrane, and the vesicles spill their contents of the neurotransmitters into the synaptic cleft. The discharged neurotransmitter molecules then diffuse across that narrow gap and bind specifically to the receptor proteins in the membrane of the postsynaptic target cell. So you can see here this little diagram, and I apologize, it's a little blurry, but it's kind of showing you that synaptic cleft. It's going to be having the neurotransmitters, and that's going to be opening up the calcium ions. So they flow in, and then that's going to fuse and spill their contents into the molecules of the synaptic cleft. And our last thing to talk about is reflexes. This is an involuntary response to a stimulus. It is going to occur automatically. It occurs rapidly because the impulse travels the shortest route possible. So it's pretty much the site of stimulus through that central nervous system. Now, a reflex arc is a chain of neurons that mediate that reflex. It begins with the receptor at the end of a sensory fiber, which then leads to the reflex center, which is a processing center where neurotransmitters, interneurons are then going to communicate with motor neurons. And just so you know, the sensory receptors are cells that respond to that change in its environment. It begins the reflex arc. And you can see here how that all will happen. And again, the POGL on the reflex arc is going to help with your understanding of that. One kind of cool thing to talk about is the stretch receptor. That is the knee-jerk response. The sensory neuron whose end is wrapped around a muscle fiber is embedded within the muscle of a thigh. When the tendon of the knee is tapped, the attached muscles stretched, activating this receptor, which generates this neural impulse. The terminal processes of the sensory neuron end in the spinal cord in synapses with the motor neurons leading directly back to the thigh muscles. So the impulse is traveling back along these motor neurons, cause the muscle to contract, and the leg extends forward. It is evolved as an adaptive response that helped you maintain your posture and balance. And it's also going to be found in mountain goats, which is pretty cool to see. Not if you actually have a mountain goat, but I think it's a pretty cool fact. Not all sensory neurons from stretch receptors in the thigh muscle terminate at the motor neuron, which returns to that muscle. Many sensory neurons synapse with interneurons of the spinal cord, which carry impulses to the brain, where we perceive that stimulus and send commands to our muscles. And again, it is the knee-jerk reaction, and it is our ability to stand